Hello, medical intervention students. This is Mr. Fall. I'm going to talk through what your assignment is for e-learning day, February 9th, 2021. So we have left off by talking about gene therapy case studies. So we were in 221. And in the workbook, we are at the cystic fibrosis case. I asked for you to get as far as you could on that yesterday. So number one priority will be to finish this as best you can. And bring back any questions you have next time we're in session, or email me questions throughout the day if you have any. Then the next step after that, I would move on to this Alec Faustus case study, where you're going to look at the situation. He essentially has some form of hemophilia, and that would be found, all that information is found here. One of the key things a lot of people miss is this button right here, click access test results. And one super important piece of information is the size of the gene. And essentially, you're going to find out information about his genetic disorder. You see that he does have hemophilia A. The bleeding disorder, body does not make this clotting factor 8 successfully. It's made in the liver, and it affects the ability of the body to stop bleeding. So you're going to develop a gene therapy strategy for Alec. And so once you have an idea of what you think would be the correct vector and the correct tissues, you're going to test that out. So you do a little bit about the pathology and how it's inherited, how the gene is altered. And then you're going to choose the correct vector and the correct tissue and justify why each is or is not a good choice for the vector and why these are the two best options for the tissue. And when you get to that, test the gene therapy strategy. If you click here, when you click on these, you can kind of do trial and error to figure it out. But the key thing is making sure they're the right size and making sure that they can, for carrying the 2600 base pair segment, and also that they are able to infect these cells correctly. So it goes back to the notes that you took over the different vectors. Hopefully you've done this section already, and can use that to help find the best vector to help do the gene therapy. After that, if you're finished with that, I would say that's where you should definitely get today, is done with this case study. So if you can get through this point, that would be great. If you're already there, then I would encourage you to go ahead and work ahead. This CRISPR paper model thing, we'll work through some of this in class, so skip over that for now. And all I really want you to do is look at this, what's called CRISPR Click and Learn. Now, what this whole system talks about, I want you to look at a couple of videos and this interactive. So if you go into courses, this is under the genome editing technology part 2, and it talks about CRISPR. What does CRISPR stand for? Clustered regularly interspaced short palindromic repeats. Of course, I'm sure you guessed that. Basically, there are little repeated segments, and a palindrome is where it's the same forward and backward, found in the genome of bacteria, where this little system was discovered. And so that, along with a restriction enzyme which cuts DNA, is used to help locate DNA sequences that need to be edited, and then to cut them to either render them useless. If it's maybe a gene or a protein that's causing a problem, you don't want to use it anymore, it can basically knock it out. Or it can be edited and replaced with something that is more useful. Okay, so as far as the videos go, I would encourage you to watch this video, this Mr. Anderson Bozeman science video. Watch that to kind of get an idea of how it works. And then from there, go to do one workbook and this last page of it, and click on this link here. All right, and when you do, it's going to take you to this interactive activity, and basically you're going to fill in the blanks and kind of summarize it. So this information here at the start will help you fill out the information, the blanks here. to understand what it does. So essentially, think of like someone has a genetic disorder, potentially this could edit and that and cure the genetic disorder. And so, summarizing the steps of that, you'll see the steps listed, and these are screenshots from the activity. And basically, if you just go back here and click on how it works, then you'll see how this works. So basically, you're going to scroll down, or basically scrolling upward, which is making it go downward. Okay, so it's like I'm moving down, or you can just scroll down on the screen, and it will give you more and more information. It's kind of guiding you through how this molecule works. So this is the CRISPR-Cas9 complex. And so basically there's 4 stages. It targets a certain sequence and it's given a guide. Each molecule can be customized, giving a guide RNA. And that guide RNA basically is going to look for a certain sequence that is what is needing to be edited, whatever the target DNA is. And so there's a little sequence called PAM. Basically, long story short, is a little sequence that is basically the landing spot for the CRISPR-Cas9. So you see this little yellow sequence here. That's where these repeat over and over and over again. And those are the different places where it can sort of land and associate with the DNA. And then you'll see the targets, and it finds that DNA that it's wanting to bind to, the target DNA, and the guide RNA binds to it. And then that's the CRISPR system doing that. And the Cas9 part is the restriction enzyme. Once it binds to it, it cuts it, cleavage site, right? Okay, so it cuts the DNA. And then DNA tries to repair itself, but it isn't the best at repairing. So it's going to create a mutation. And what actually happens is the sequence gets cut again and again and again and again. So even if it repairs it once, eventually it's going to be incorrect. And that's essentially what's called knocking out a gene so that it can't be used anymore. So there's really like two, sorry, wrong thing there. It can knock the gene out to where it really isn't useful anymore. And so that's one way of doing it is basically knocking the gene out so it doesn't function because of this mutation. Or a homology-directed repair is basically adding in a DNA repair template to the mixture, I guess, whenever you add the CRISPR-Cas9 system into it, so that whenever you put overabundance of this repair template, then when this gets cleaved, the repair mechanisms are going to insert this correct sequence that hopefully makes it work in a more functional manner. So basically, long story short is I don't need you to turn this in yet. This is not due till Friday, so for now, work through the cystic fibrosis and the hemophilia alk-Faustus case studies, and then skip over the paper model thing for now, and just do the CRISPR click and learn, which is on the last page, using the link that is right here. There's also links in Canvas that you'll see right here. And so don't do anything with reproductive technology just yet. We'll see when we meet again and all that. Just make sure you have these things turned in, like the Judy Smith, the journal visits, the 213 and 215 you should have turned in on paper, and all the most recent assignments. All right, that is it. Let me know if you have any questions. Send me an email, and hopefully I'll see you soon. Thanks.